Hello everyone. Today we will come to know about the basic features of a human eye, and in abridgment through this video we all will be acquainted with the basic procedure, means how the human eye helps the human body to see everything. Through this diagram we will come to know, and later we will come to know about the basic defects, means myopia and hypermetropia, with the correction procedures. So let's make a start with the basic diagram, basic features, structural features of a human eye. So let's have a look through this diagram. This is a basic sectional diagram of a human eye. And this part is the front, this part is the posterior portion. This portion is conjunctiva. Beneath conjunctiva we will notice this is cornea, and the posterior portion of cornea is sclera, and beneath sclera this portion is retina. And this is the lens. See what lens is placed. Okay, this is iris fiber, and through this fiber lens is hanged. The anterior portion of the lens, the humor is known as the liquid, is known as aqueous humor. This portion is filled with vitreous humor, and this is a blind spot. From this spot the optic nerve is generated, and what happens, this basic feature, functional feature, how human eye helps us to see. This lens is natural convex lens, and light rays, this incident ray, okay, light rays comes from this medium, is the outer side, and through this lens, this is a convex conserving lens, natural, and after refraction the refracted rays are conserved, so they are conserving in this way. This is a point of focus. And in this diagram, just compared to this, light rays comes from a medium, this lens, and after refraction the refracted rays are converged through the process of converging, okay, and it is focused at this point, and this signal is propagated through the optic nerve to the brain. The signal is transmitted to the brain, so we can see everything, and this is the basic feature, structural and functional feature of a human eye. The retina is composed of two types of cells, rods and cone cells. So through rod cells we can see the difference between the light and darkness, and by the help of accommodation we see in the time of dark and at the time of light, at daytime and night time. Through the help of this rod cell and cone cells helps us to identify the colors, means red, green, blue colors, and different types of color. Identification can be helped by the use of these cone cells. So these are basic functional features of the retina. And the next point is the defect with correction. Two types of defects we will know about through this video, and one is myopia, which is nearsightedness, and another is hypermetropia, which is far and long sightedness. And what is myopia? Suddenly what happens, the person's eyeball, the person's eyeball becomes larger, and the process is different totally. And just have a look through this diagram. Normally what happens, the incident rays are refracted if the eyeball is enlarged, so the refracted rays are conserved at this point of focus, and which is totally far from the retina, the spot where normally the image, the refracted rays are conserved, the image is produced at this point, but this point is totally far from this point, and this is actually in between the vitreous humor. As a consequence, what happens, this image, the actual object is placed in this position, and we notice without the help of specs, we notice the object is placed far from this position and is totally blurred. Okay, so the refracted rays are conserved at this point, the focused at this point in between the vitreous humor, in spite of focusing at this point on the retina. What happens, the image that formed in this way, the object is placed far from this original position. So we notice the object is totally blurred. This type of defect can be corrected by the use of a concave lens, means in front of a natural convex lens we have to put a concave lens, and by this process the actual refracted rays can be converged and focused at the point of focal point and the point of retina, and by this process the image is actually, we can notice the object is placed in its original point, and this is in this way this defect of myopia can be corrected. Okay, and the next point is hypermetropia. Here the eyeball is suddenly decreased, and what happens, the light rays comes from a median, and through this natural convex lens the refracted rays are converged and focused behind the eyeball. Okay, the actual image should be produced, the converging process must be done at the point of retina, on the retina, but in the patient of a hypermetropia patient, what happens, the image is formed, the refracted rays are converged, the focal point is behind the retina, behind the eyeball at this point. So this type of defect leads to the, what happens, the image is placed in this point, but we notice that the object is placed near the eyeball, means this what happens, if the object is placed in this position, we notice the object is placed near us. Okay, so this type of defect should be corrected by the use of another convex lens. This convex lens in the front side of a natural convex lens, and through this use of convex lens, the actual image is formed on the retina and the object is totally placed in its original position. So hypermetropia defect is corrected by the use of a convex lens, and myopia problem is corrected by the use of another concave lens. Okay, this is the main difference between two types of defects, means myopia and hypermetropia, and this is how these two types of defects are corrected. So I hope the foresaid discussion is going to be useful to your study. Stay tuned to YouTube channel Knowledge Press. Subscribe the channel. Happy learning. Thank you.